Today, we're going to talk about sickle cell disease and CRISPR. Sickle cell disease affects nearly 100,000 Americans as red blood cells, the carriers of oxygen from the lungs to the body, start to form into sickle cell shapes, which stick together, restricting blood flow and causing symptoms like acute and chronic pain in oxygen-demanding conditions. How is sickle cell disease developed on a molecular level? Red blood cells contain millions of oxygen-carrying molecules called hemoglobin, which contain 4 subunits that each hold an oxygen molecule within an iron heme structure. The hemoglobin is present in 2 states: the T state, where the heme is domed and has limited oxygen binding affinity, and an R state, which forms a planar heme which allows for higher oxygen binding affinity. Well, humans also have 2 types of hemoglobin based on different stages of human development, controlled by transcription factors: fetal hemoglobin, or HbF, at 0 to 6 months of age, and adult hemoglobin, HbA, at 6 months to death, replacing the 2 gamma globin subunits with beta globulin. Well then, how is a sickle cell disease phenotype generated? What occurs during sickle cell disease is that there's a spontaneous mutation of the HBB gene on chromosome 11 that produces beta globulin, which can occur from DNA polymerase errors or spontaneous chemical changes such as deamination. Here, an adenine is replaced by a thymine. Although such small point mutations often have little or no effect to what occurs later in translation, this is a missense mutation because it leads to a change in the amino acid added during translation, where glutamine is replaced with valine, leading to hemoglobin S, HbS. Unlike glutamine, which is hydrophilic, valine is quite hydrophobic, and therefore causes 2 major changes in the hemoglobin structure. In high oxygen-demanding conditions, such as after a person does exercise, the hemoglobin will enter its T state, exposing hydrophobic sites such as the valine. This valine is then attracted to a hydrophobic component of another HbS, called the E pocket, and binds to it. This HbS has also an exposed valine that can bind to a different E pocket. This leads to a quick polymerization chain of HbS's that reduces oxygen binding affinity, induces cell shape changes, and results in early death and the symptoms of sickle cell disease. Now, what are some ways that scientists are trying to solve sickle cell disease? One promising and creative way that scientists are trying to tackle sickle cell disease is by using CRISPR-Cas9. Some researchers at Harvard have found a gene, BCL11A, that produces a transcription factor that represses the formation of HbF after development. They've used CRISPR-Cas9 and a specific guide RNA complementary to the gene to bind, create a double-strand break, and use non-homologous end joining to create a mutant gene that silences the expression of BCL11A and allows the body to produce more HbF. Remember, HbF doesn't have a beta globin domain, which means it doesn't form polymerization and replaces the sickle cell blood cells with ones that are normal. Clinical trials have shown that a majority of patients who receive this treatment have little to no symptoms of sickle cell anemia. And now that's the story of amazing science. That's it for today.